In this last section, we're going to discuss eukaryotic photosynthesis. This is a simple representation of a photosynthetic organelle, the chloroplast. An organelle is a small molecular machine inside a cell. So what are we putting in? We're putting in sunlight, carbon dioxide, and water, and we're getting out glucose and oxygen. An improvement over prokaryote photosynthesis occurred when cyclic electron transport and the storing of energy in the bonds of carbon atoms were combined to create non-cyclic electron transport. Chloroplasts use this process to produce all sugars and carbohydrates. So we're going to take a closer look at this process in the slides to come. Another improvement, so in addition to non-cyclic electron transport, another improvement is that we don't just have one photosystem, but we have two that absorb light and create ATP called the light reactions of photosynthesis. They take place in the thylakoid membrane of the chloroplast. The Calvin cycle or dark reactions then create glucose with the energy from ATP. So you can see we have two photosystems here, one involving light reactions and the second called the Calvin cycle. So light reactions take place in the thylakoid membrane of the chloroplast, which you can see labeled here. The absorption of light from the sun, the creation of ATP and NADPH, those three things together are called the light reactions of photosynthesis. Okay, they take place in the thylakoid membrane inside the chloroplast. So what's happening? We're absorbing light, creating ATP and NADPH. Those are the light reactions. Light-dependent reactions convert solar energy, so energy from the sun, into high-energy molecules, ATP and NADPH. So we're in the thylakoid membrane here, creating ATP and NADPH. The Calvin cycle, or the dark reactions, are light-independent, and that is where we're creating glucose. Carbon dioxide processing. Carbon dioxide is processed by plants in three different pathways at the beginning of photosynthesis depending on the environment and the type of plant. So the C3 pathway is the pathway that occurs under normal conditions for most plants. The C4 pathway occurs under high temperature, high water, and high light availability conditions. Okay, so plants that exist under those conditions and is a slightly more efficient pathway for photosynthesis. And then this CAM pathway, CAM stands for crassulation acid metabolism. This crassulation is a type of plant that this pathway was first identified in, and it occurs in plants that exist in high temperature, low water environments. So like in the desert, we have this CAM plants process. C3 is used by most plants, and those plants can also use C4 when necessary, or CAM when necessary. CAM is a subset of C4, so they're never used by the same plant, okay? Photorespiration is a wasteful pathway that occurs when the plant takes up oxygen and releases carbon dioxide in the presence of light, reducing the effectiveness of photosynthesis. In this case, the Calvin cycle enzyme rubisco acts on oxygen rather than carbon dioxide. C3 and C4 photosynthesis. The C3 process is used by rice, wheat, and potato. C4 photosynthesis is about 50% more efficient than C3 and is used by maize, sorghum, and sugarcane. Only 3% of all plants use the C4 process. C3 plants have no special features to minimize photorespiration. In other words, it's a less efficient process because there's no features to minimize this wasteful photorespiration. Okay, so in the C3 photosynthesis, you can see on the left here, we're undergoing this Calvin cycle in these mesophyll cells. And you can see that that's slightly different than what happens in this C4 photosynthesis. So we have this extra step here, this extra feature here to make the C4 photosynthesis a little bit more efficient. Let's take a look at that process now. Plants that are in hot, dry environments use the C4 process to minimize photorespiration that occurs at the surface of the leaf. Two different parts of the leaf are involved as the carbon dioxide uptake process is split into two different types of cells. The light-dependent reaction, mesophyll cells, is separated from the Calvin cycle that is performed in the bundle sheath cells. So you can see we have the two processes separated, one process occurring in the mesophyll and one process occurring in the bundle sheath cell. CAM photosynthesis. CAM plants, including succulents such as cacti, agave plants, grapes, and orchids, minimize photorespiration and save water by separating these steps in time between night and day. So like plants in the desert, they don't want to lose all of the water out of their stomata. They want their stomata to be closed during the day. But the problem is that photosynthesis only occurs during the day and needs carbon dioxide. So plants have evolved this CAM process where they've separated the steps between night and day to make it more efficient. So the plants absorb carbon dioxide in the nighttime, fixing it in an organic acid with the assistance of an enzyme, and release it in the daytime where it undergoes the C3 process to create glucose. Plant species that use CAM photosynthesis not only avoid photorespiration, but are also very water efficient. Their stomata only open at night when humidity tends to be higher and temperatures are cooler, both factors that reduce water loss from leaves. C4 and CAM plants are found in dry, hot environments as they conserve water better than C3 plants. Light-dependent reactions occur in membrane-bound structures called thylakoids. The thylakoid membrane separates the inside of the thylakoid, or the lumen, from the outside, or the stroma of the thylakoid. So you can see we have the lumen on this side. This is the inside, and this is the stroma. This is the outside. So you can see that's where light is coming from outside. The inside is called the lumen. The outside is called the stroma. And of course, we're producing ATP here and NADPH here. And we're sending those to the second step, the Calvin cycle, or the dark reaction. All right, so let's take a closer look here because we said there were two photosystems. So light-dependent reactions use light energy and water to form ATP, NADPH, and oxygen. NADPH is formed by adding a proton to NAD+, storing energy in that addition, in that bond, that NADPH. The process requires two photosystems, each of which is a collection of proteins that absorb light and transform that energy into energetic electrons. Photosystem 2 occurs before photosystem 1 because it was discovered first. Here is the net reaction after both photosystems operate. We have 2 water molecules, 2 NADP plus molecules, 3 ADP, and 3 phosphate groups. And we produce an oxygen, 2 NADPH, and 3 ATP, right? That's what we're forming in these light-dependent reactions. Photosystem 2 is the first stage of photosynthesis. Light energy from the sun is absorbed by the chlorophyll molecules in the chloroplast energizing electrons. You can see that here, light energy to photosystem 2. The energy splits a water molecule that releases oxygen to the atmosphere. So we need water, right? So that's one of our reactants, and we split it, and then we have oxygen and then the hydrogen ions. The net result of the reaction is 1/2 O2 plus 2H plus ATP. This reaction yields the oxygen that we breathe. So let's take a look here. This is photosystem 2 right here. The chlorophyll is absorbing energy from sunlight, energizing electrons, and splitting a water molecule. So here comes our water molecule right here, and we're splitting it into 2H plus 1/2 O2. The oxygen is emitted to the atmosphere. All right, now let's talk about these electrons. When the light is absorbed, the electrons in photosystem 2 are energized, right? The energized electrons move through the thylakoid membrane. This is known as an electron transport chain, transferring their energy to membrane proteins called proton pumps. So you see these protons moving across the membrane this way. The motive force of those electrons moving in this electron transport chain gives the energy required to move these protons across the membrane from outside in. Proton pumps actively transport, so it's active transport of protons. That means energy is required. And again, where does the energy come from? The energy comes from the movement of the electrons in this electron transport chain. Proton pumps actively transport protons, hydrogen ions, against their concentration gradient from outside, from the stroma, inside into the lumen. There are now more protons in the lumen inside of the membrane than in the stroma outside, resulting in a net positive charge in the lumen and a net negative charge in the stroma. The higher electric potential in the lumen should cause those lumen protons to diffuse to the stroma where the proton concentration is lower. So you'd expect them to move back along with their concentration gradient. But the semi-permeable membrane prevents that. Instead, we need a channel protein, and so that's provided by ATP synthase, this enzyme. It provides a channel back to the stroma. It provides a pathway for those protons to move outside and a catalyst for the ATP creation reaction. So protons move through ATP synthase via facilitated diffusion. The final movement of the protons uses their electric potential. So when they're inside of the membrane in the lumen, they're sort of like at the top of a hill. And as they move through the ATP synthase channel, they fall down the hill. And in doing so, that provides energy or potential to do some work in the form of electric potential energy. And what that electric potential energy is used for is adding a phosphate group to ADP, creating ATP. Electrical energy in this case is converted to chemical energy. Electrical energy from the protons moving from high electric potential to low electric potential, converted to chemical energy stored in the bonds between the two neighboring phosphate groups. 3 molecules are generated for every molecule of oxygen formed. And this process of converting electrical energy to chemical energy is known as chemiosmosis. At this point, the electron has lost most of the energy it had originally acquired in photosystem 2. In order to carry out the rest of photosynthesis, the electron needs to recharge. The electron reaches a second chlorophyll molecule located in a protein complex called photosystem 1. There, chlorophyll absorbs more light energy and re-energizes the electron. All right, so these re-energized electrons here combine with NADP plus, forming NADPH, okay, through the addition of one of these protons here. NADP plus is the terminal electron acceptor in photosynthesis. Overall, the reactions in photosystem 1 transform light energy into chemical energy, NADPH. The net result of the reaction is one NADPH and one proton for each water molecule that is broken up. So remember that this comes from the water molecule, where we split the water into 2H plus and half O2. All right, so here's what happens at photosystem 1. So here's our electron. Remember, it's used up most of its energy at this point. The electrons from photosystem 2 that powered up ATP synthesis as they pass down the electron transport chain have to be re-energized by light. So there's a more light input at the chloroplast here in photosystem 1. Once these electrons are re-energized here, they are used by the enzyme NADP reductase to create NADPH and H plus, a proton or a hydrogen ion. So remember, we talked previously, we have this NADP plus and 2 protons. The enzyme works to attach one of them to this NADP plus so that we have NADPH and a remaining proton right here. And why do we do that? The purpose is for storing energy. So the energy is now stored in that NADPH. And we have energy available there to do work. All right, so let's go back to this diagram that we saw before. This is the end of our light-dependent reactions. After photosystem 1 is finished, it passes the ATP and the NADPH to the Calvin cycle, also known as the light-independent or dark reaction. Some of the sun's energy that was absorbed by the chloroplast has been stored in those two molecules, ATP and NADPH. All right, let's take a closer look at the Calvin cycle. The Calvin cycle can occur in light or dark. Thus, dark reaction is not an accurate name, but it is traditional. The cycle takes place in the stroma of the chloroplast, which is the aqueous fluid that exists outside of or in between the thylakoids. The Calvin cycle uses the energy stored in ATP and NADPH to convert CO2 into glucose in a multi-step process. All right, so let's take a closer look at this Calvin cycle. And this looks really overwhelming. There's a lot going on here, but I'm going to go through each step of the process overall and then describe what we have summarized here. So remember that the overall process of the Calvin cycle is to convert carbon dioxide, CO2, from the atmosphere, so we have CO2 here, to carbohydrates or sugar or glucose, which the plant needs to power its activities and do other things related to the structure of the plant. The Calvin cycle reactions, remember, they occur within the stroma of the chloroplast, outside of the thylakoids. The reaction is using the ATP and the NADPH produced by the light reactions to convert the carbon dioxide into carbohydrate or glucose. So there are three main phases in the Calvin cycle: carbon fixation, carbon reduction, and then regeneration of the RUBP. The first phase, the carbon fixation, captures the carbon dioxide from the atmosphere so that it can be used in the reactions. How are we capturing the carbon dioxide? Well, we're attaching it to this RUBP molecule, which is a 5-carbon molecule. And we use the enzyme rubisco to do it. So you see we have this 5 carbon molecule. It interacts at this enzyme to capture a CO2, which is a 1 carbon molecule, right? So you see the addition of that carbon there. This is an unstable 6 carbon molecule that quickly splits into 3 carbon molecules, 3-phosphoglycerate, which you see labeled here as 3-PGA. The next step in the cycle is reduction, or the conversion of that CO2 to carbohydrate or glucose. This is CO2 reduction because we're adding electrons and energy to the CO2 molecule. There are a sequence of reactions that use the ATP and the NADPH from the light reactions. 6 each, 6 ATP, 6 NADPH. These ATP and NADPH supply the needed electrons and energy for carbon dioxide reduction. Electrons are added from NADPH, and through a series of reactions, this 3-PGA is reduced to form G3P, which is a carbohydrate. The ATP is now ADP, and the NADPH is now NADP+. Those are returned to the thylakoids to be converted back to ATP and NADPH by the light reactions. One G3P molecule is set aside to make glucose, and most of the rest, the remaining, continue in the Calvin cycle, this third phase of the Calvin cycle, or regeneration. In this regeneration phase, ATP is used to combine the rest of the G3P molecules to form a new RUBP molecule. Again, back to that same 5 carbon sugar that we need to be able to capture the carbon dioxide. This RUBP can combine with additional carbon dioxide molecules, continuing the carbon reactions. To form glucose, the cycle has to turn 6 times because each turn of the cycle adds only 1 carbon atom from the incoming carbon dioxide. So I've summarized on the right here. In 3 turns of the cycle, only 1 carbon dioxide molecule enters the cycle at a time. The cycle uses 3 carbon dioxide, 9 ATP, and 6 NADPH, and makes 1 3-carbon sugar, G3P. We need 2 of those to be put together to form glucose. Okay, so that means in total, the cycle has to turn 6 times in order to get 2 of those 3-carbon sugars to form glucose. Remember that the Calvin cycle is dependent on the light reactions, which provide the NADPH and ATP, which provide the electrons and energy needed to form the carbohydrates. The Calvin cycle is also called carbon fixing. Carbon, a gas in the atmosphere in the form of carbon dioxide, enters photosynthesis in the Calvin cycle. It is turned into a solid as glucose. When glucose is used in cellular respiration, the next topic, that carbon is released back into the atmosphere. The process of fixing and releasing carbon is called the carbon cycle. Carbon is not being created or destroyed, but is being cycled throughout the environment. All right, let's take a look at our photosynthesis roadmap here and just kind of summarize everything we've talked about in this section, because it has been a lot. Photosynthesis consists of 2 separate series of chemical reactions that capture electromagnetic radiation and transform it into stored chemical energy. Light reactions occur in the thylakoid or on the thylakoid membrane. They require light. They use water, ADP, and NADP+ to produce oxygen, which is released to the atmosphere, ATP and NADPH, which are both then moved to the Calvin cycle, the second series of reactions. So the Calvin cycle occurs in the stroma, in the space outside of the thylakoids. The Calvin cycle creates sugars using carbon dioxide and the from the atmosphere and the ATP and NADPH from the light reactions. ADP and NADP+ are released as byproducts, and those, of course, are then returned back to the light reactions so that the cycle can continue. Photosystem 2 is the first structure in the series. Light excites the pigments of the photosystem, and electrons are sent to the electron transport chain. Simultaneously, water molecules are being split and providing their electrons to the photosystem. Water is the primary donor of electrons. Electrons are not returned to the pigments. This is a non-cyclic electron transport here. The electron transport chain moves electrons as described in cyclic electron transfer, but the electrons are no longer excited because of the energy transfer. They are then donated to the next step. Photosystem 1 and photophosphorylation are our next step. Photosystem 1 re-excites the electrons using light energy. Remember, they've moved from photosystem 2. That motive force worked to move some protons, but now they've used their energy. They have to be re-excited. These high energy electrons are used to reduce a molecule of NADP+ to NADPH. This will carry the electrons to the second stage of photosynthesis, the Calvin cycle. Finally, the proton gradient is used to generate ATP. The enzyme ATP synthase takes advantage of the energy of the hydrogen ions' natural diffusion down its gradient. The energy is used to attach a phosphate to ADP. The energy harnessed from sunlight has powered the generation of ATP. This is known as photophosphorylation. The light-dependent reactions are the first part of photosynthesis. The remaining reactions are called the Calvin cycle or the light-independent cycle. The Calvin cycle uses NADPH and ATP produced from the light-dependent reactions to reduce carbon dioxide and produce sugar. This is called carbon fixation. At this point, photosynthesis is complete. Light energy combined with carbon dioxide and water formed glucose and oxygen. Much, but not all of the sun's energy has been transformed and stored as chemical energy in glucose. The reaction will run in the opposite direction within living organisms, including the plants that made the glucose, to extract the energy in glucose to run their life processes. We will learn how living things extract energy from glucose in the next unit, cellular respiration.